Hello and welcome. I'm Juliana Ranches. I'm the host of RancHER and I'm assistant professor and beef extension specialist for Oregon State University. I'm based in Burns at the Eastern Oregon Agricultural Research Center. And at OSU, my job is split between research and extension. And with that, it's with great pleasure that I bring to you all the 4th edition of RancHER. So tonight we have two great speakers and I'm very excited for their talks. We have Dr. Clara Menezes and Dr. Cecily Bishop joining us. But before we start, I wanted to remind you some of the housekeeping, not rules, but how we go with the program, right? So for questions, the only way to interact with our speakers is through the Q&A box. So if you have questions, you can ask those questions during the presentations using the Q&A box, which should be at the bottom of your screen if you're connected using a computer, okay? We will address those questions at the end of each talk. So Dr. Menezes will go first, she'll give her presentation, and then we will address those questions, and we'll follow with Dr. Bishop, and at the end of her presentation, we'll address questions related to her presentation. Another reminder is that as every year, we do have a door prize. So if we stay with us to the end of the night tonight, you'll be running for the door prize. We have a $100 Amazon online gift card. So that will be there will be a draw for that at the end of the night, okay? So I think those are the main recommendations I have for tonight. So without any further ado, it's with a great pleasure that I introduce the first speaker of tonight, Dr. Clara Baiao Menezes. Dr. Menezes is assistant professor of ruminant nutrition in the Department of Animal Sciences at South Dakota State University. The two main focus areas of Dr. Menezes' research program are fetal programming and digestive physiology. Dr. Menezes aims to optimize dietary formulation for pregnant cows and heifers by better understanding the maternal-fetal crosstalk, as well as the impacts of dietary treatments on nutrient digestion, absorption, efficiency of energy utilization in beef cattle. Dr. Menezes obtained her bachelor's in animal science from Federal University of Viçosa and obtained her master's and Ph.D. degrees from the same university. She spent part of her Ph.D. program at the North Dakota State University as a visitor researcher. And she did a postdoc in nutrition and reproductive physiology at North Dakota State University, where she investigated the effects of maternal vitamin and mineral supplementation on fetal development of offspring efficiency of energy utilization in beef cattle. Tonight, Dr. Menezes will be presenting nutritional strategies for pregnant heifers and cows. Anna, thank you very much for joining us tonight, and the screen is yours. If you want to share your video and share your screen, that would be great. Thank you.
Thank you, Juliana, for the introduction. So I'm going to share here my presentation with you. Let me know if everything is okay. Can you see the screen?
Yeah, we can see it well and hear you well. Thank you.
Okay, perfect. Well, thank you for the introduction. Before I get started, I would like to give you a little background about me. I'm from a small family farm in Brazil. My dad, he had a mixed operation with a few dairy cows. So I knew since I was a kid that I wanted to work with cattle. So I went to college. I got my bachelor's in animal science by the Federal University of Viçosa, and I got my master's and PhD by the same institution. During my master's and PhD, the major focus of my research was energy and protein requirements and metabolism in finishing cattle. And during my PhD, I was awarded a scholarship. So I got to spend 1 year of my PhD program at the North Dakota State University. There, I worked with Dr. Joel Caton, and I was introduced for the first time to the concept of fetal programming. So I started assisting in projects where we evaluated the effects of nutrient restriction during gestation and its impacts on the offspring. So when I finished my PhD, I was invited to continue at NDSU as a postdoc researcher. So I spent 2 years at NDSU as a postdoc, and my projects were focused on maternal vitamin and mineral supplementation during gestation and its impacts in the offspring. So in 2022, I was hired at South Dakota State University. So I actually never left the Dakotas. I just moved south. And here at SDSU, my two major research areas are fetal programming and nutritional physiology. So with that, let's jump right into the main point of this presentation, which are nutritional strategies for pregnant heifers and cows. Well, the first study that I'm going to be presenting was performed at North Dakota State University during my postdoc, and the major focus was mineral supplementation to gestating beef heifers and their impacts on fetal programming. The second study is currently being conducted at SDSU, so I will be presenting some preliminary data regarding two different winter feeding strategies that we are testing, either a high forage versus a high concentrate diet, and their effects on methane emissions, as well as colostrum and milk quality and production. And to wrap up things, we'll have a short take-home message. In this first study, as I mentioned, the major focus was mineral supplementation for gestating beef heifers and their impacts on fetal development or fetal programming. So I'll get started with a quick background presenting the main ideas or the main questions that led us to the development of the studies. On the first study, we focused on supplementation pre-breeding up to the end of the first trimester of gestation. And on the second study, we supplemented the heifers throughout the whole gestational period, and we evaluated the impacts on the F1 and the F2 offspring. So we evaluated the effects on the daughters and the granddaughters. So it was a transgenerational programming. Well, this graph represents the major fetal developmental milestones during gestation. And I would like to catch your attention for the events that take place on the first trimester of gestation. So this is a period of intense metabolic activity where critical factors take place, such as maximal placental growth, differentiation, vascularization, organogenesis. So during this period, the fetus requires an adequate supply of essential nutrients. And among those essential nutrients, the minerals, they play a key role in several biological and physiological functions, such as hormone and DNA synthesis, cellular antioxidant defense, membrane electric potential, nerve transmission, metabolism of nutrients, catalysts and enzyme systems, among several other functions. And the maternal intake of nutrients is the major factor that will impact the transport or the supply of minerals to the developing fetus. And inadequate maternal intake of nutrients may have long-lasting negative impacts on the performance and health of the offspring. But despite the importance of the first trimester of gestation, despite the importance of maternal nutrition during those early stages of gestation, most producers, they tend to focus supplementation at the last trimester of gestation as a strategy to raise body condition scores prior to calving. And also this late gestation period is where we have an increase in the nutrient requirements of the pregnant cow. So we have exponential fetal growth, and we know that those animals, they are going to be demanding more nutrients. So most of the research that we have done so far regarding fetal programming is also focused on the last trimester of gestation. But there are several different nutritional strategies or supplementation strategies that are adopted by producers. And in terms of mineral nutrition, we have not only several different sources of minerals like organic, inorganic, injectable minerals, but also there is a wide variation about whether or not to provide mineral supplements to replacement heifers, like throughout gestation or in specific stages of gestation. So those were the basis for our first study. And the major question that we have in this research project was, does maternal mineral supplementation in early gestation affect fetal development or fetal programming during those early stages of pregnancy? So here we worked with a 2x2 factorial study. So the first factor was vitamin and mineral supplementation. So we are going to be abbreviating here VTM as the animals that were supplemented with vitamins and minerals, and no VTM for the animals that did not receive supplementation. So this VTM supplementation started at least 60 days prior to breeding, and they were supplemented up to day 83 of gestation. Our second factor was the rate of gain. So here we worked with two different rates of gain, either a low gain of 0.29 kilograms a day or a moderate gain of 0.79 kilograms a day. And this moderate rate of gain was achieved via protein and energy supplementation. And the rate of gain factor started being applied at breeding and went up to day 83. So day 83 of gestation was our endpoint. So here, all of the heifers, they were ovariohysterectomized. So we removed the gravid uterus and we collected the placenta and the fetus. We dissected the fetus and we collected and measured all of the fetal organs. But for this presentation, we are going to focus on liver, muscle, intestines, and brain. So we used a commercial supplement, okay? So it was inorganic mineral sources. Here you have the composition of the supplement that we used. So the macro as well as the trace minerals. And this supplement also provided vitamins. So vitamin A, D, and E. All of those heifers, they were individually fed. And we used the dose that was recommended by the manufacturer. So it was 4 ounces per heifer per day or 113 grams per heifer per day. So I put together this table where we can see the mineral composition of each one of the treatments that we had, okay? So this represents the amount of minerals considering the basal diet plus the supplements, if recommended by treatment. And here, to your right, I added the NRC requirements so we can see how our diets compared to those. And even in our control diet, so the heifers that did not receive any supplementation, they were marginally deficient in some of the minerals, for instance, copper and zinc. So with that, one of the first things that we tried to understand was the relationship between mom and fetus. So mom and fetus, they communicate to each other through the placenta, okay? So to evaluate better this crosstalk or to try to understand this communication, we ran a differential gene expression analysis in the caruncles, the maternal side of the placenta, and the cotyledons, the fetal side of the placenta. So the results are presented here in those two images. So on figure A to your left, we have a differential gene expression in the caruncles, the maternal side. And on the right, we have a summary of the genes that were differentially expressed in the cotyledons, the fetal side of the placenta. And overall, we observed that we had a greater number of genes that were overrepresented in the caruncles compared to the cotyledons. And also, we could see, as I highlighted here in red, that whether providing supplementation or not resulted in alterations in the expression of genes. But the most important question is, what are the biological pathways or the biological functions that are controlled by those genes? And in the case of the caruncles, we observed upregulation of genes related with hormone synthesis and lipid metabolism, while on the cotyledons, we observed an overexpression of genes related with nutrient transport and biosynthesis of amino acids. And those differences between maternal and fetal side, they are expected because on one side, we have the fetus that is demanding a great supply of nutrients. And on the other side, we have the mom that is trying to make the most out of the resources that she has available to supply those nutrients to the fetus. Okay, but did the changes in placental gene expression affect the nutrients available to the fetus? Yes, they did. So we observed a greater concentration of trace minerals in fetal liver, muscle, and allantoic fluid for those fetuses whose moms received supplementation in those early stages of gestation. So in fetal liver, we observed a significant greater concentration of selenium, cobalt, copper, and manganese. In fetal muscle, we observed greater concentrations of selenium and zinc. And in the allantoic fluid, we had greater concentrations of selenium and cobalt. And another interesting result was the fact that the liver and the intestine weights were greater for those fetuses from VTM heifers compared to no VTM heifers. And I'm going to be exploring this a little bit more later on this presentation. So in addition to those differences in mineral concentrations, we also observed that independent of treatment, there was always a greater concentration of minerals, especially selenium, copper, and zinc in fetal liver compared to maternal liver. So this tells us that the mom is prioritizing the transport of minerals to the fetus. We also observed a greater concentration of amino acids in the allantoic fluid of VTM heifers. So 12 out of the 14 neutral amino acids, they were present in greater concentration in the allantoic fluid of VTM heifers, as well as histidine and aspartate. And we also observed that the most abundant amino acids were glutamine, glycine, and alanine. Well, glutamine is one of the most important fuels for fetal growth. So this amino acid can cross the placenta and it can be converted to glutamate. And glutamate will then enter the TCA cycle to generate energy, to generate ATP. Glycine is the most abundant amino acid in the repro tract, while alanine is considered a gluconeogenic amino acid. So it is used to generate energy, to generate ATP. Okay, so far we saw that supplementation of minerals during early gestation altered the expression of genes in the placenta and also altered the transport of nutrients to the fetus. But how can we measure or how can we observe impacts on fetal development? So to dive more deep into that, we collected samples of fetal tissues and we ran a differential gene expression analysis. So we observed that several genes, they were either up or down regulated in fetal liver, muscle, and brain. So in fetal liver, the genes that were upregulated were related with mineral metabolism. That was kind of like expected, since we saw that there was a greater trace mineral concentration in the liver of VTM fetuses. We also observed an upregulation of genes related with the transport of amino acids, which makes sense since we also saw that we had that greater concentration of amino acids in the allantoic fluid, and we saw an upregulation of genes related with lipid metabolism. So for the sake of time, I am not presenting here the data that we got regarding lipid metabolism, but we conducted a lipidomic analysis in fetal liver, and we were able to identify over 500 lipid metabolites. And we observed that mineral supplementation definitely played a role for some of those lipid metabolites. So in fetal muscle, mineral supplementation resulted in an overexpression of genes related with tissue growth and development, while in the brain, we had an upregulation of genes related with energy metabolism and brain development. Okay, so as a summary, so far we saw that maternal nutrition, pre-breeding, and up to day 83 of gestation affected placenta gene expression, increased the concentration of trace minerals in liver, muscle, and allantoic fluid, increased the concentration of amino acids in allantoic fluid, and we also saw that there was differences in the expression of genes in fetal liver, muscle, and brain, okay? But what does that mean? Will the offspring of supplemented dams be more efficient? So this led us to our second study. So in this study, we provided supplementation to the heifers throughout the whole gestational period. And one of the major questions that we had was if the supplementation, the maternal supplementation, would affect feed efficiency and efficiency of energy utilization in the offspring. So for this project, we worked with 2 cohorts of heifers, okay? So this was the first cohort. So we applied the treatments from breeding up to calving. So we had only 2 treatments here. So the animals were either supplemented or not with this vitamin and mineral blend. It was the same commercial supplement that we used in the first study. We slaughtered the calves after birth, and we collected samples of all of the organs, and we took several measurements as well. So we did not observe any significant differences in calf birth weight, as well as in the weight of the organs, as you can see here in this table. So even though numerically we did have heavier liver and small intestine for the VTM fetuses, that was not considered significant, VTM calves, sorry. So after slaughter, I collected samples of liver and intestines. So remember that I previously showed you that liver and small intestine of those fetuses in early gestation, they were heavier, right? So liver and small intestine, they are the major drains of maintenance energy in cattle. So I wanted to study more in depth what was going on in terms of energy metabolism in those organs. So I subjected those samples to a high resolution respirometry analysis. In a nutshell, basically, I was trying to figure out how much ATP was being produced in the mitochondria of those organs. So you're gonna see that this graph right here is similar to the one that you're gonna be seeing on the next slide, okay? So here we have on the yellow bars, our control treatment, so the animals that were not supplemented, and on the green bars, the animals that were supplemented, or whose moms actually were supplemented. On the y-axis, we have the oxygen flux or the oxygen consumption that can be correlated with the synthesis of ATP. And on the x-axis, we have the respiratory stages. So represented here by L, we have the leak respiration. So that means energy that is lost as heat and is not used for biological functions. P is the oxidative phosphorylation. PI is the oxidative phosphorylation supported by glutamate. and E is the electron transfer capacity, okay? So here, contrary to what I was expecting, we did not observe any differences between our treatments for production of ATP or efficiency of energy utilization in the liver. However, when I analyzed the small intestine, so the jejunum samples, we did observe significant differences for all of the respiratory stages that we studied, okay? So this greater synthesis of ATP in the jejunum may indicate a greater efficiency of nutrient absorption and utilization. But will those animals perform better, gain more weight? So that's when we had our second cohort of heifers. So again, the treatments were similar, okay? So we provided or not vitamin and mineral supplementation from breeding up to calving. However, here we followed up the offspring. So we followed those F1 heifers all the way through the development phase and throughout gestation. So those heifers, they were bred, and during the whole gestational period, we subjected those animals to a series of tests, I would say. So we used the head boxes as an indirect calorimeter approach to estimate energy requirements and also to evaluate methane production. We did liver and muscle biopsies on those animals with the goal of also understand more about energy metabolism in those tissues. And at the end of the last trimester of gestation, those heifers were slaughtered, and we collected the F2 fetus, okay? And we are currently analyzing this F2 fetus. So first, I'm going to be presenting some results from the F0 heifers. So the heifers that received the treatments. We conducted a series of liver biopsies over time in those animals. The first liver biopsy was performed at breeding, so at treatment initiation, and then we performed biopsies at the end of the first trimester of gestation, day 84, at the end of the second trimester of gestation, day 180, and at calving. So here to the left, we have the concentrations of selenium, and to the right, the concentrations of copper. So the white bars represent the control animals that were not supplemented, and the black bars, the animals that were supplemented. So as we can see, for both copper and selenium, there was no significant differences between the treatments at breeding, which was expected, since that was our starting point. But as gestation advanced and as those animals were receiving their supplements, then we had significant differences between those two groups, where the heifers that received vitamin and mineral supplementation, they had significant greater concentrations of selenium and copper than the heifers that were in the control treatment. But for both groups of heifers, we observed that there was a decrease in the hepatic concentrations of selenium and copper as gestation advanced. So at calving, we also biopsied the calves. Okay, so here we can have a comparison between the concentration of those trace minerals in the moms and in the calves. And as we can see, the cows, the heifers, they were prioritizing the transfer of nutrients to the calves. So even those animals that were not being supplemented, that were deficient, they still prioritized the transfer of nutrients to the calves. But why should we worry about trace mineral reserves at birth? Well, one of the major reasons is because milk is not considered a good source of trace minerals. On this table here, I highlight the NRC requirements for calves and the amount of trace minerals that are provided by the milk. And as we can see, the milk is not going to be able to supply the amount of trace minerals that are required by the calves. Okay, so that's why it is important that those animals, they are born already with mineral reserves, okay, in their livers. We also studied the effects of our treatments on colostrum production and colostrum quality. And those heifers that were supplemented throughout gestation with that vitamin and mineral blend, they have greater production of colostrum, and they also had a greater concentration of trace minerals in the colostrum. So this was a very interesting finding for us. And as I mentioned, we followed those offspring throughout the development phase. And even though we did not observe any differences in birth weight, we observed significant differences later on. So at 6 months old, those animals, they were weaned, okay? And we can see here that always the VTM offspring were heavier than the control offspring. So the VTM is being represented here by this solid line, and the dashed line represents our offspring from the control dams. Okay, so we can see that here also I would like to highlight that those offspring, they were managed under the same conditions. Okay, so no treatments were applied to their moms after calving or to those calves. So we are assuming that all of the differences in body weight that we are observing in here, they are an effect of the fetal programming. So the nutrition that those calves received while they were in utero. And I am very passionate about placenta and the transfer of nutrients throughout gestation. So of course we collected placenta samples at birth after they were expelled. And we evaluated the vascularization of the placenta. So we could see, so we could study like how we could correlate better that supply of nutrients with the transport of nutrients to the fetus. So I added those two images here. The top image, figure A, shows the placenta of a control cow. And on the bottom, figure B, it's the placenta of a heifer that received vitamin and mineral supplementation. And all of the red dots that you see, they represent the blood vessels. So we had significant greater vascularization in the placenta of those heifers that were supplemented throughout gestation. But to unravel better this relationship between placenta vascularization and transport of nutrients, we are currently performing gene expression analysis in the cotyledons, so we can better understand this relationship. So as a summary, so far we saw that supplementing heifers throughout gestation affected energy metabolism in the small intestine of calves. So we saw that there was a greater synthesis of ATP and that those animals, they would be more efficient. We saw that it resulted in greater concentration of trace minerals in the liver, in the colostrum. That also resulted in heavier offspring. Okay, so even though we didn't observe differences in birth weight, we did observe differences later on throughout the development, plus placenta vascularization was also affected. And as I mentioned, those heifers, they were, the F1 heifers were slaughtered at the end of the last trimester of gestation. So now we are analyzing any possible epigenetic mechanisms so we can see if those effects, they would be inherited. Okay, so now we are studying if the treatments that were applied here on the grandmothers, if they are going to be observed in the granddaughters. We are not done with all of the analysis yet, but I am very excited and I'm looking forward to see if we are going to see any differences, especially in energy utilization in those two targeted organs like the liver, and the small intestine. So with that, let's move on to our second nutritional strategy. So this is preliminary data of a study that is currently being conducted at South Dakota State University. We are evaluating two different winter feeding strategies, either a high forage or a high concentrate diet. So for a quick background, winter feeding costs, they typically represent the largest portion of a cow-calf operations expenses. And here in the Midwest, usually the winter time often corresponds to the last trimester of gestation and early lactation, which is a period where we have an increase in the nutrient requirements of the cows, but we don't have enough pasture. So we need to extend sometimes the dry lot period, and we are going to be adopting different winter feeding strategies. And there is no one size fits all when it comes to winter feeding strategies. However, here we decided to explore more about using high concentrate corn-based diets and see how that would compare to a conventional high forage hay-based treatment. So we applied our treatments starting 2 months pre-calving up to 84 days post-calving. So we had a high forage group and a high concentrate group. Our high concentrate group was limit fed, so they received 1.4% of their body weight. And our major goals were to evaluate the effects of those strategies on colostrum production and quality, milk production and composition, methane emissions, and calf performance. So here is the table with our experimental diets. When we formulated those diets, we targeted them to be isonitrogenous, so the same amount or a similar amount of crude protein between those diets. And what varied the most here was the energy content. So the high concentrate diet had a greater energy content than the high forage diet. You can see here that we have 70% of dry rolled corn included in this diet. And here on the high forage diet, we had greater proportions of hay and corn silage. And both groups were supplemented with vitamin and mineral liquid supplement, providing more than 100% of the NRC requirements to avoid any potential deficiencies. Those animals, they were housed in a group pen equipped with electronic feeders and electronic waters. So for those of you who are not familiarized with the Incentec feeders, this is an electronic system. So basically it measures by difference how much the animal ate. All of the animals, they are fitted with an electronic ID tag. In the ear, we can't see well here in this image, but basically when the animal approaches the feeder has a sensor that will read the tag and then will allow access to the animal. And this system is connected to a software, so we can control how much that animal is allowed to eat in a day. There is a small gate here in this system that will allow or not access to the animal. So if this cow, for instance, already ate her daily allotment, the feeder is not going to allow her to enter and eat more. So that's how we were able to control the intake of our high concentrate cows, and we were able to keep them on their targeted 1.4% of body weight intake. We used the green feed system to measure methane emissions. So the green feed system is also a type of electronic feeder, but it works a little bit different. So here we use pelleted food as a bait to attract the cows. And the green feed feeders, they have systems that will be measuring the concentrations of methane and CO2 that are eliminated via eructation when those animals are eating. And we also receive like real time the data about the productions of methane and CO2. We are very interested in milk quality and milk production. So my students, they had lots of fun milking those cows every 2 weeks. So we milked them from birth up to day 84. So that's when we stopped applying the treatments. So those cows, they were fully milked. So we used portable milking machines. We adopted a separation protocol between cow and calf of 12 hours, and they were completely milked. And we analyzed not only milk yield, but also milk composition. This first table here has the results regarding colostrum. So I didn't have a better picture, but what I tried to show here is that right after birth, we would bring the cow and the calf to the chute. We would keep the calf right here in front of the cow so she could calm down, and then we collected the colostrum. We collected colostrum from the left rear quarter. So all of the measurements that you have here, like the volume, they were based on how much colostrum was being produced in that quarter. And here we have the concentrations of the nutrients. There were no significant differences between high forage or high concentrate cows, even though numerically we can see that the high concentrate cows had a greater concentration of fat, protein, and lactose. When we evaluated the milk production, though, then we had significant differences between our treatments, with the high concentrate cows producing more milk than the high forage cows. And we also have the nutrient composition of the milk throughout this period, including weaning, because we kept on milking those cows even after we were done applying the treatments. But this would be just like a huge table to add to this presentation. But we observed that the nutrients, either in their absolute value or when we corrected for the volume of milk, there were no differences between our treatments. And regarding the methane emissions, as expected, the high forage cows, they produced more methane compared to the high concentrate cows. And that was a constant throughout the project. So here, this graph shows the data that we got from treatment initiation. So this was in January 15th when we started. And then here is when we ended our treatments. So it was pretty constant that those high forage cows, they were producing more enteric methane than the high concentrate cows. And here I just separated that data between the pre and the post-calving periods. So pre-calving period here to the left, figure B, and on figure C, we have the post-calving period. But again, as you can see, high forage cows, they had greater methane emissions than the high concentrate cows. So as a summary, so far, we observed that high concentrate cows, they have increased the milk production and reduced methane emissions. As I mentioned before, I am really into the placenta and trying to analyze and understand what's going on in there. So we collected placentas of those cows, and we are currently analyzing gene expression in the placenta so we can see if we had any differences in the expression of nutrient transporters, more particularly glucose transporters, since we worked with those two very different energy levels in our treatments. We already performed the immunoglobulin analysis in colostrum. We did not observe any differences regarding concentrations of IgA, IgG, or IgM. And we are currently on the final stages of developing an online economic tool for producers. And this tool is going to be housed at the SDSU Extension website. But my master's student, Megan Warbine, she's the one responsible for this trial. She presented some preliminary economic data at the Minnesota Nutrition Conference. And based on current prices of feed ingredients, as well as the prices of hay that we had on the last winter, we could see that adopting this corn-based diet in this restriction intake can save money to producers. I believe that in our calculations, we could save $28 per cow. So, but again, we are still working on refining our system. And then the idea is that we are going to have this interactive platform housed in our website so the producers can actually input data from their own operations, and they can see if those strategies are going to be helping them to save some money or not. So with that, my major take-home message is that what mom eats matters. From those trials, we could see that maternal mineral supplementation affected placental function, organ development, offspring body weight, and feed efficiency, and that winter feeding high concentrate diets on late gestation and early lactation increases milk production and reduces methane emissions. But to figure out what are the mechanisms behind the responses that we observed, how they are modulated, and we also should explore more how we can take advantage of fetal programming. So with that, I would like to acknowledge all of the students, faculty, and personnel from North Dakota State University, South Dakota State University, and Auburn University, as well as the funding agencies that made this research possible. And thank you, Juliana, one more time for the invitation. I was really looking forward for the RancHER. And with that, I am going to open for any questions that you may have. Thank you.
Thank you, Dr. Menezes. That was an amazing presentation. You really touch on, I work with mineral my entire life, trace minerals specifically, and now I'm working with methane too. So I was really also looking forward to hear your presentation. I think you're doing amazing work. You share a little bit of the transfer of trace minerals, and I was looking at your data. I was like, on your control cows from day 84 to the calving, it's so fascinating to me that there's no change and they still have minerals to provide to those calves, right? Like they were all I don't know how it's a miracle, but it always mesmerized me, like, to think about that. I really appreciate the data though. I do have a question, but we do have a lot of questions on the Q&A box if you want to go ahead and read out those. But I was just curious to see on your methane data, do you have a comparison between pre and post calving methane emissions? It was something that just crossed my mind and I'm curious about it.
Oh, okay. Let me just go back here. Okay, so those graphs right here.
Yeah.
Okay, so we have the methane emissions over time throughout the whole period. But here I just kind of like divided them between the pre and the post calving period. But of course I had to work with an average of the calving date. So I do this analysis.
Yeah, I think it would be, I mean, there's differences in the intakes that I would expect to see differences on the methane emissions. And just looking at this data here, there might be something there. So I think it'll be interesting to see. Our cows here are all dry cows, so I don't have the opportunity to look at the data. But anyway, I don't want to take more time, the time of the attendees. So if we want to go and do the Q&A box questions, there's quite a bit of questions there.
Okay, perfect. Oh yeah, we have a lot of questions. So let me see here. The first question is from Tom. Please explain up and down regulation of genes. So Tom, I'm not like a geneticist by any means, so I'm gonna try to explain the better that I can here. But basically, upregulation of genes is gonna be showing which genes are gonna be present or expressed in greater amount, in greater number. And downregulation is the genes that would be expressed in lower amounts. So that's how we can study how they are differentially expressed. So some are going to be upregulated, so they're gonna be more expressed, and some are downregulated, so they have a lower expression. So that's kind of like the way that I would explain it. The next question is from Jocelyn. Are there any other benefits to using the green feeds to see the methane production in your study other than feed differences? Yes, Jocelyn, and that's a great question. So not for this project specifically, but for a follow-up project that I'm going to be doing now next year, we are going to be using the green feed also as a tool to estimate energy requirements. So for this next trial, we are going to be applying the exact same treatments that we are doing now, but we are going to be doing a digestibility trial as well, and we will be collecting urine samples. So if we have the concentrations of methane plus the concentrations of nitrogen in the urine, we can use a equation, a Brouwer's equation that it's called, so we can estimate the energy requirements of those animals. So my major goal with that is to evaluate differences in the requirements, energy requirements of those cows on late gestation and in that early lactation period. There was a paper that was published in 2023 that validated the green feed as a tool to estimate energy requirements. So that's one of the things that I want to see better how it works. I'm fascinated by energetics. So what I really would like to do is actually a comparison between the head box system and the green feed and see how they actually compare to each other. So that is my major goal, like something for the future, but that I really would like to do. Next question is from Erica. Thank you for the presentation. What are your next goals for advancing methane emission studies? I kind of like answered your question already on this other one, but yeah, I really would like to do this comparison between the head box system because we have more, it's not completely closed circuit, but we can have measurements over a 24-hour period that would be maybe more accurate than the green feed system that is completely open, but I don't know yet. That's kind of like one of my goals for the future. Let me see here. I kind of like lost where I was.
Sorry, and I did just check that one down,
Oh,
but
okay.
it was basically like what would be the next steps or studies on methane emissions and how to reduce methane emissions. I'm sorry, I was just checking them. Sorry.
Oh, okay. Perfect. We have a question here from Andrea. Regarding your first trial, did you observe any differences in conception rates of the F1 heifers based on the supplementation of F0? Great question, Andy. We did not observe any differences on that. So we did ultrasounds and we measured the size of the ovaries. We measured the number of follicles. No differences on that. We collected blood samples for progesterone concentrations, and also it was similar for the heifers from both groups, like for the F1 offspring, sorry, from both groups. It was exactly the same. Then we have a question from Shelby. In the first experiment, were there any significant environmental or health challenges to the calves? If not, would you expect the same outcome if there were significant challenges? Shelby, no, there were no significant environmental or health challenges. We did those trials in a facility that was very similar to the one that I had here in the pictures. So it was in a barn, a controlled environment. We were keeping an eye on those animals the whole time. So they were not challenged, but it would be very interesting actually to do some sort of health challenge to those animals, like expose them to pathogens somehow and see how their immune response would work, because we know that trace minerals, they are closely associated with the immune system. So that would be something really interesting to be done. That's a great idea. Beth, she's asking if we can transfer this information to sheep production. Beth, this is a very interesting question. So even though sheep and cows, they are both ruminants, they are different species. So I would use some caution when trying to completely translate the data that you see here for sheep. The ideal would be conducting a similar trial, like either the mineral supplementation trial or this energy gestation trial in sheep, so we can see if the responses can somehow compare or be related to each other. But I would use some caution when trying to translate results from cows to sheep. Do you know a range of time when the economic extension tool will be available? We hope that by the end of December. So we hope that by December 31st, that tool will be available in our website. Then we have a question from Tom. Fetal tissue respiration. How does increased respiration relate to efficiency? Tom, in that case, we are talking about cellular or mitochondrial respiration. So we were relating the mitochondrial oxygen consumption with the efficiency of utilization of substrates to generate energy. So the efficiency in which the electrons transport in the transport chain are actually being able to produce energy and not being wasted as heat. So that's how we could correlate that animals that would be more efficient in coupling this transport of electrons with synthesis of ATP, that those mitochondria are more efficient, and consequently, we hypothesized that the small intestine that we collected to evaluate would also be more efficient in the utilization of nutrients. And when we observed that the offspring on that developmental phase was actually heavier, we kind of like think that we could correlate both things. Then we have a question from Riley. In the first study about mineral supplementation, were the heifers managed on dry lot or on pasture during the summer after AI? On dry lot. So they were managed in a dry lot setting.
Okay, I think those are all the questions. Very good questions and very good answers. I appreciate that, Anna. Thank you very much. I know that you might have to go, so I just want to express my appreciation for you joining us tonight. I really, really enjoy your presentation. I want to say to the attendees, if you have any questions later, and if Anna's not longer here in the meeting, she has a little one waiting for her at home. So if she's not here and you have questions, I'll make sure to pass the question along to her. So we'll get that addressed at some point. But thank you very much. Really appreciate you joining us. I think you're doing an amazing work. And I'm looking forward to see the continuation and the results of all your trials. I think you're doing great. Thank you, Anna.
Thank you so much, Juliana. And we should keep on touch, so then we can talk a little bit more about this whole like mineral deal.
Yeah, absolutely. That would be fun. Thank you.
And thank you.
Okay, so now moving to the second half of our night, we'll learn from Dr. Cecily Bishop. Hi, Cecily. So Dr. Cecily Bishop is assistant professor in the Department of Animal and Rangeland Sciences at Oregon State University in Corvallis. So we are department mates, but she's in Corvallis. I'm here in Burns. Cecily received her bachelor's degree in animal science from Washington State University in Puma, Washington, and her Ph.D. in animal sciences biology of reproduction with a minor in biochemistry and biophysics from Oregon State University under the mentorship of Dr. Frederick Stormshack. After receiving her Ph.D., she completed her postdoctoral fellowship under the mentorship of Richard Stauffer in the Division of Reproductive Science, which is now the Division of Reproductive and Developmental Sciences at the Oregon National Primate Research Center at Oregon Health and Science University in Beaverton. She transitioned into a staff scientist and research assistant professor before joining the faculty at Oregon State University as assistant professor. Her research group at OSU has explored the use of 3D follicle culture techniques to induce antral formation in cattle follicles, a Guinea pig model for polycystic ovary syndrome, and the use as a model for ovarian cryopreservation and retransplantation, which I believe is probably my favorite study, actually. I think it's really cool. She has also worked with vitamin supplementation to improve fertility outcomes in cattle. Cecily is a member of the American Society of Animal Science, the Society for Study of Reproduction, the Society for Reproductive Investigation, and American Society for Reproductive Medicine. She currently serves as the chair of the Federation of American Societies for Experimental Biology Science Research Conference Study Advisory Committee. Tonight, Dr. Bishop will be presenting strategies for preparing low-parity heifers and cows for their next breeding season. Thank you, Cecily, and the screen is yours.
Great, thank you, Juliana. Okay, so I am, as Juliana said, I've had a lot of variations in my career, but I am really happy to share some of the research that we've been working on and also some strategies that both myself and several other colleagues have worked on to prepare low parity heifers and cows for their next breeding season. And in fact, I'm going to use most of my time in this talk to actually walk through some concepts that have been developed over the years and to highlight some really nice tools that are now available to the community from especially the Beef Reproduction Task Force. So with that, I will get started with my talk. So just because the last talk was very nutrition intensive, we're kind of switching gears a little bit. We're still going to talk about nutrition because it's very critical to the process for fertility, especially in our younger heifers and cows. But I wanted to just introduce some concepts just so everyone's on the same page. So this is the estrus cycle in cattle and bovines. So if we look at what is going on hormonally, and so as reproductive biologists, we spend a lot of time thinking about the hormones and the structures that are releasing them. So the major hormones that we are talking about for cattle and fertility are estrogen, progesterone, luteinizing hormone, and follicle stimulating hormone. And estrogens come from the development of a follicle that contains the egg or the oocyte. And as it grows and it gets ready to ovulate, we get a release of a large amount of estrogen from that follicle that causes estrus. And then those levels fall as the follicle ovulates and is reorganized into the corpus luteum. The corpus luteum is actually incredibly critical. Many of you will know produces progesterone to maintain gestation. So if pregnancy occurs, progesterone levels should stay high. If pregnancy does not occur, in a cow, progesterone levels will fall and she should come back into estrus in another 21 days on average. So estrus in our cattle can be incredibly short. So we're talking about a period of time for standing heat where that cow is receptive for breeding between as little as 6 hours to up to 24 hours. The average is about 18 hours, but it will vary by breed. Now, estrus, occurs first, that receptivity, that breeding event, but ovulation actually doesn't occur until later. So ovulation and actually rupture of that follicle containing that egg, that oocyte, that's able to be fertilized, that is actually not going to be released until 24 to 32 hours after the start of estrus. And that becomes really critical, especially when you're trying to do some of the managed breeding programs. So if you are trying to use artificial insemination and you're trying to do some of the less expensive synchronization programs that rely really heavily on being able to breed at a detected estrus, we've got a very small window to work in. And I actually have a link here in this presentation. And there's going to be a QR code later on in the presentation to some of these resources for the Beef Reproduction Task Force. They actually have a really great heat detection planner that can work through options. There are several options for heat detection to make sure that we're breeding at the right time. So now to the first calf heifer. So the first calf heifer, this is her first cycle. I am going to start arguing to all of you, and I hope by the end you're going to agree with me that we are actually setting up this heifer for her breeding almost from the time that we select her as a replacement heifer. And so the reason this becomes important is because we need to set that heifer up to be able to get pregnant very early in our calving season. If she is not able to get pregnant earlier in the calving season, she is going to be less likely to become pregnant and have an offspring in subsequent calving seasons. So it becomes a domino effect. If she does not get pregnant right away, as many of you know, as many of you may have experience with, she is not going to be able to calve in time to be able to recover from that calving, be able to breed again and have another calf that is the same in your same breeding season. And this is actually some data from a fairly old study at this point, where they actually looked at the number of cows that failed to calve the next breeding season, and how far along into the actual calving season they calved. So if it was a first calf heifer and she was due for her first calf, if she calves in the first 20 days of your calving season, so for many of us, we like to talk about winter calving. In my area, we're talking those cows that are going to calve hopefully by the end of January, beginning of February. If they calve early in the season, the chances are really high that they're going to calve again. They're going to have their next calf in our chosen breeding season their following year, so their second calf. So in these data, only 7% of those cows failed to have another calf in the next breeding season. And as you can see here, as you go through, as she calves later into your calving season, she is less and less likely to have that second calf in a timely fashion. You can hedge your bets here by choosing heifers, your replacement heifers that are born themselves very early in this calving season. So if you're choosing older replacement heifers, those heifers that are usually greater than about 398 days old at the time of your breeding season, they're going to have a really high pregnancy rate. They're going to have a 90% pregnancy rate that first breeding season. If you even go down to even 20 days younger, the data suggests that you're going to get at least a 4% drop in pregnancy rate. But if you stretch that out to 42 days younger, it can be as low as 76%. Reduced pregnancy rate. And again, you know, if you have a larger operation, if you have 100 replacement heifers, that is, you know, quite a big chunk of your heifers not getting pregnant in that first season. So if you can help it, choose the older ones. You may not be able to. You may have some of your replacement heifers. You want to be replacement heifers that have pretty good genetic merit. Again, you may not be able to help this, but you can help yourself if you're choosing an older replacement heifer. But also with choosing that replacement heifer, again, hedging your bets a bit, you can determine if they're ready for breeding by using a reproductive tract scoring system. And so this is typically done by your veterinarian or another trained individual with palpation. It's usually best if it's done 30 days before the start of your breeding season, before you want to be breeding this animal. And the score, for those of you that are unfamiliar with this, it's a score of 1 to 5, where 1 is the animal is still prepubertal, the tract is very immature, and there's no ovarian activity. We don't feel follicles. We don't feel a corpus luteum present. And then it goes up to 5, where that is a female that is completely ready for breeding. Her tract has gone through puberty. We can feel that in the tone of her tract. She's going to have a more what we call turgid uterus. So her uterus is gonna feel more meaty, and we can identify a corpus luteum present. And again, this is some really nice data that I really like from a review of some colleagues of mine, where they looked at several of these studies. It didn't matter whether the breeding was done by natural service, so that's NS, if these were synchronized females that they performed artificial insemination on, or if this was just judged at the end of the breeding season. Those females with higher reproductive tract scores at the start of pregnancy, even in the Bos indicus, which are generally a little bit more difficult to synchronize and breed. If you have any cows that have some Bos indicus genes or genetic background, they're all of them that have that higher reproductive tract scores are more likely to get pregnant than their counterparts with lower scores. And again, that's pregnant overall in the breeding season, but again, we know that they're also going to be the ones that are usually going to get pregnant on that first artificial insemination synchronization or earlier in a natural service. So hopefully we've got a mature replacement heifer, or at least that she has a mature reproductive tract. So now, what about preparing for that second breeding? So again, you should think about that second breeding when you're preparing to breed her the first time. So we know that pregnancy rates per cycle for heifers born from mature cows, as well as older replacement heifers, appear to be higher in their second breeding season versus a less mature first calf heifer. And notice how big that drop off is. So some of this can be circumvented. So you can use some advanced reproductive techniques. In those first calf heifers, you can use flushing, you can use oocyte pickup to produce embryos from those younger heifers that you then transfer to your recipient cow. That seems to help rescue this fertility phenotype that we're seeing. But we can also help that heifer again when we're planning her first breeding. So we're kind of planning for her second breeding at the time of her first breeding. So if we look at how long it takes a cow or a first calf heifer to return to estrus after parturition. So that's during that postpartum interval, that time from calving to their first estrus. Notice there's a huge range. Mature cows, however, should come back in. They should be resuming their cyclicity and able to be rebred between 30 to 90 days. That first calf heifer, it's not unheard of for them to take about 120 days. So again, if you're looking at the days postpartum, and we're looking at those individuals that we are trying to breed, so ideally they are cycling, they are having their estrus cycles, Those, if we can wait until they're at least 90 days out postpartum, a higher proportion of them are going to be cycling. Again, your first calf heifer is going to lag behind, but giving that first calf heifer the opportunity to calve earlier in the calving season is going to give her then a little bit longer of a time to go through that postpartum interval, undergo a process that we call uterine involution, so that she can then be prepared to be bred again early in our breeding season. So again, just to summarize, to kind of hit it home, so to prepare for that second breeding, we nearly need to do this at their first breeding. Ideally, we're picking the older replacement heifers if it's possible, but we're definitely using the reproductive tract scoring system to confirm their ability to breed. And we're breeding early in our chosen breeding season, and we're preparing for this longer postpartum interval. This is just kind of baked into that first calf heifer. It takes them a lot longer to bounce back. We can also, again, help that first calf heifer out by avoiding dystocia. So we can select bulls with good calving ease scores for artificial insemination or natural service. So two of the EPDs that you're going to want to look at in beef are the calving ease direct, and hopefully many of you are familiar with these, or the calving ease maternal. Again, that's in making sure that our lines have good calving ease. While they're getting their reproductive tract scored, we can actually do pelvic scoring at the same time. However, a proper body condition score can avoid issues. So if she has a body condition score in beef cows of at least a 6, at the time of calving, she is going to be able to avoid dystocia. And then if you are trained to intervene if labor is prolonged, and that is more than 1.5 hours. So we know that every 30-minute delay in assistance adds about 6 days to the postpartum interval. Personally experienced this myself in a lot of our reproductive projects here. I'm going to talk about one project that we are currently underway with at the end of this. But yes, we have definitely seen this, and knowing how to assist, being trained on assistance so that you can get in there and make sure that that cow or that first calf heifer successfully delivers the calf. I'm going to do a big plug here for our wonderful extension folks around the state, organize different calving schools that can teach you the skills, at least so that you can handle some minor dystocia where you don't have to call the vet out every time your cow is having difficulties in labor. If they're just short, easy to correct. So in that body condition score, and again, we've seen this, and I know others have talked about this in the RancHER, so I'm not going to belabor it too much, but I really want to emphasize it, that that BCS of 6 before calving is really important, especially to that first calf heifer, because we want her to then maintain her body condition after calving. So ideally, she has a body condition score of over 5 after calving. Again, that's going to really help that cow to recover. She's going to be more likely to cycle. And again, I'm showing some data here, but notice how that first calf heifer, she is more sensitive to that body condition score. So if her body condition score at breeding, again, this is when we're looking at rebreeding that female, we need her body condition score to be at least a 5 to have a decent chance that she's going to have her own estrus cycles and have recovered from calving. So having a really good nutrition program is really critical to fertility, and I cannot emphasize it enough. And then also use an effective synchronization protocol. So again, I am showing you some resources that the Beef Reproduction Task Force has made freely available online. And here is that promised QR code here, where you can find all of these different protocol sheets that really show you in kind of graphical form how these different synchronization protocols work. And these synchronization protocols are going to be slightly different if we're talking about a heifer or if we're talking about the cow, the female that has had multiple pregnancies. So in our heifers, we are always going to want to include a progestin. So that's either in the form of a cedar insert or in the form of oral progestin. And that is because we need that progesterone to help that female show a really good estrus. So pre-exposure and pre-treatment with progesterone before ovulation is very critical to showing that good estrus behavior. When we see that good estrus behavior, we actually know that cow or that heifer has a follicle that's producing a lot of estrogen and hopefully is going to ovulate us a really nice high quality oocyte that's going to have a high chance of getting fertilized. So when we look at these different protocols, you're going to notice that there are some that have heat detection. So again, you're going to want to have a good estrus detection program. You can use estrus detection aids, and there are several listed here. Or you can do what we call fixed time AI. Hopefully, if several of you are familiar with this, if you are not, this is where we have a combination of hormones exposures where you inseminate the animal on a schedule. Typically, the fixed time AIs are going to be slightly more expensive per cycle than the classic, what we call heat detection in AI. Again, depends on the one you use, but they're also going to have a lower pregnancy rate. Because the nice thing about using a heat detection aid is those animals that come into estrus early, if you're using a heat detection aid, you can then breed them appropriately when they come into estrus. And then if you do not see them come into estrus, you are prepared to induce ovulation. In animals that aren't showing estrus, it may be that they just have a follicle there that has not produced enough estradiol to show a really good sign of estrus, or maybe there's other issues why you miss the estrus. It gives you a little bit of an insurance, and you can make a note on that in animals that did not show estrus before you bred them. Those are ones that personal experience and several other colleagues that do this. Those are ones that are more likely to be bred by your cleanup bull. So I have starred one here. This is one that we can use in both cows and heifers. This is my current favorite one that our research group uses. We have a really nice, decent pregnancy rate with this one. With our heifers, we average usually between 75 and 80% on a good year pregnancy rate following artificial insemination, their first cycle, as well as our cows. And those females are usually then synchronized, and if they do not get pregnant their first cycle, it seems that the cleanup bull gets them their second cycle. So this one involves uses of prostaglandins, and prostaglandins are used to lyse the corpus luteum that is present. We give a shot of prostaglandin and then we use a heat detection aid because that can, if a female is suitably mature and cycling, that alone is going to induce follicular development, and the female will ovulate by herself, and we don't really need to do a lot more here. So we use the estrus detection aid. Any female that has ovulated, shown estrus, we are going to artificially inseminate those females, and we actually save ourselves some of the remainder of the protocol. So we could save ourselves some money there. In females that did not show estrus, we then give a shot of gonadotropin releasing hormone, or GnRH. Again, if those females just did not have a good enough follicle to give us enough estrogen to show estrus, that will cause that follicle to prematurely ovulate and form a corpus luteum. Can also synchronize the follicle waves so that we are more likely to get a dominant follicle later in the cycle. We insert a progestin device. We like a cedar, and this is a vaginal device for those that are not familiar with it. We then give these females a shot of prostaglandin 9 days later. We are going to lyse any CL that may have been present. And we also remove the cedar, and we use an estrus detection aid. And again, we like the Estrotec patches. They're kind of, for those of you that are not familiar, they're like scratchets almost that go on top of the tail head. When they're rubbed off, you know the female's in estrus. And then we heat detect for 72 to 84 hours. During that time, any female that comes into heat, we will breed by artificial insemination. Those females that have not come into heat by 84 hours, we're going to do that final shot and perform artificial insemination. So in this protocol, in our hands, we're going to find that most of our mature cows, about 50% of them are only going to need that first shot of prostaglandin if they're a more mature animal. Those that needed the cedar, again, about half of those are going to show, if it's early in our breeding season that we use AI, half of those are going to show estrus on their own, and then they're not going to need that last shot of GnRH. So, and again, there are lots of different ways to go about this. So this is going to depend on your manpower, your labor, your setup. But that is also, again, where, and I'm going to keep plugging it, the Beef Reproduction Task Force does a really nice job of showing different options, and they can even consult. There are some people available for consultation. So, and then I have, if you have not considered synchronization for your herd, they also have a estrus synchronization planner. And let me see if I can pull this up. And you should be seeing a worksheet now. And so this is a worksheet where I can list out what type of system do I have, what type do I want to do estrus detection, do I want to do just fixed time AI. There's some different options. I use, I've put in fixed time AI just for the sake of demonstration. My breed type, I have mostly Bos Taurus. I'm using conventional semen. And my synchronization protocol is I like that prostaglandin 6-day cedar with a fixed time AI. So I've put in the protocol that I want to use, and then my date to start breeding and the time I'm breeding. How many and when I'm going to turn in my animals. So right now, to minimize labor, we turn in the bull really early. In our research herd, we just want them pregnant. I'm going to say that I would say you're going to want to wait if you want to know who the parent is, if your animal is by AI or by your cleanup bull. So, and I have my group size, and what it will do then is it runs it through. I have 30 head in my group. It's telling me my labor estimate is about 27.7 hours with this protocol. And it's about my labor charge. I did not change this, but you'll want to change it to your going wage. And then it has also, if you want to calculate how much forage your animals might be consuming during this system, and then my cost of my drugs and my semen. So it's going to be able to tell me what is my cost per protocol. And that is going to be, I can change these down here. I haven't done this. So again, this tells me a little bit about the system that I chose, that it works well in cows. I don't really require estrus detection, but I will. It can initiate estrus cycles in some non-cycling females. That's why I like it. But I'm going to expect lower fertility if my cows are less than 50 days postpartum when I start them on the protocol. It also gives me what day I am to do what. So I should start, if I want for my breed date, I should start April 15th. What am I going to do that day? Tells me what drugs I'm going to do throughout the entire protocol. So it's really nice. It also gives you this in a calendar too, if that's how you like it. So it's a really good resource, and I wanted to share it because I know that the Beef Reproduction Task Force is hoping that this gets picked up and really widely used. So that is the Task Force Synchronization Planner. So with that, that is the tools that we have on hand right now. So I will also tell you there's a lot of really active research going on in this. And so I'm going to share with you some newer research that my group has been working on, again, to help with the idea that we're preparing that first calf heifer for her second breeding. And this time our group is going to target during gestation. So this is a project that we've been working on for a couple years now. We are looking at using a supplementation of both vitamin A, as in beta carotene, and vitamin D together during these winter months to prime reproductive success. And we use both gestating beef cows and heifers, but we've really focused majority of our effort on that heifer. I am ably assisted in this project by our Soap Creek Ranch manager, Mike Hammerich. I cannot do anything without him. He is absolutely amazing with our cattle. And my graduate student, who is our Oregon State University Beef Council Fellow, Amanda Fox. She just defended her master's and earned her master's degree as of this fall and is carrying it on these studies to her doctoral degree. So why do we care about these vitamins? So vitamin A, this is the beta carotene precursor. It's converted in the liver to retinoic acid. Majority of the studies on vitamin A have actually been looking at either fetal loss or fetal abnormalities, or in males, looking at the very well characterized role that retinoic acid plays in sperm production. For vitamin D, there's less that has been done, but there's an emerging field right now that we are acknowledging that vitamin D may be important to female fertility. This is your majority of your vitamin D is going to be through UV light exposure. But you can also get this through direct dietary intake, mostly through vitamin D3 supplements. So we kind of joke around a little bit, myself and some of my colleagues, we're all in the Northwest, so we don't get outside a lot when it's rainy and icky in the winter. So we're mostly walking around as vitamin D deficient in the summer, or I mean the winter. We recover in the summer. And deficiencies associated with vitamin D are your muscles and your skeletal disorders. Rickets is one very common one. So when we look at the NRCs, so the NRC requirements for beta carotene for vitamin A assume that all beef cattle get enough beta carotene from consuming fresh spring grass. Fresh spring grass has a very high concentration of beta carotene, and at least in adult cows, we know that it appears to be able to be stored for 2 to 4 months in the liver, but these stores can be rapidly depleted based on the energy demands of the animal. And the nice thing, at least from our perspective, if we're looking to supplement it, is actually the toxicity levels are pretty high. So yes, you can have toxicities, but you're going to have to give that cow quite a bit to see toxic levels. It's not the same for vitamin D. Vitamin D, a gestating beef cow is going to need about 125 units per pound of feed. That is based off of the idea that we just need to prevent deficiency. We know that typically during the summer months, our cows, depending on if they're housed mostly in sheltered or outdoor housing, are going to have anywhere between 20 to 50 nanograms per ml vitamin D if we measure this in their blood. However, they have a toxicity at between 200 to 300 nanograms per ml of vitamin D, and this actually results in hypocalcemia because of the close relationship between vitamin D and calcium flux in the bones. So it has been assumed that for beef cattle that all of their vitamin D requirements are met by sunlight, that they're at least not going to be deficient. But we really haven't done a great job until recent years in defining what is the absolute requirement, not just to prevent disease, but to really give a benefit to the animal. So we did some preliminary studies, and actually the in vivo part of it, the in-cow part of it, we teamed up with a dairy to do some really limited studies looking at their vitamin D levels and some fertility. But we also took and we collected oocytes, cumulus oocyte complexes from ovaries of cows at beef plants. And we took those cumulus oocyte complexes and we fertilized them in vitro. And when we fertilized them in vitro, we noticed that when we exposed them to both vitamin D as well as vitamin A, that the percentage of unfertilized oocytes when we added vitamin D into vitamin A was rapidly diminished. So we were seeing a real benefit. When we had vitamin D alone, the embryos tended to be better quality at the end of our culture period. We did not transfer these into recipient cows. This was a limited pilot study, but it provided us some really interesting data that we decided to try and get down to the bottom of and see if we could use this as a benefit to beef cow fertility. The same time that we were mulling over the start of this project, some researchers at University of Florida, Lincoln, Nebraska, as well as Minnesota looked at vitamin D levels of beef cows and calves. And they looked at 3 different herds. They had a herd in Moscow, Idaho. They had a herd in Grand Rapids, Michigan, and a herd down in Florida. So the interesting thing is all three of these herds were consuming a mineral oral premix supplement that had vitamin D in it. And when they looked at the maternal vitamin D levels, even though, and that's shown on this bottom axis here with the cow serum vitamin D, that even though the cows appeared to be fine, their calves, and especially those calves born in Idaho during their spring calving season in late winter, were had such low vitamin D levels that they were almost they should have been considered deficient. So we looked at that and thought, okay, maybe also with treating the cow, we might be able to provide some benefit to the calf through supplementation. And maybe we should do a little more direct supplementation rather than just an oral supplement, because we know that not every cow will eat the supplement equally. So we embarked on this first part of this pilot study. These cows on this study, we had a majority of them were first calf heifers and another group of second calf cows. Again, these were low parity. And we had relatively modest numbers. Again, this was more of a pilot study. And we took and we injected them monthly with an injectable that contained a combination of vitamin D and vitamin A. And that was the only supplements it contained. And we chose the doses that we did because it allowed us to raise vitamin D levels. We proposed it would raise them to about 50 nanograms per mil based on some preliminary data we had. We treated these animals monthly until they calved. And again, once they calved, we also took a blood sample from the calf within 12 hours of birth. All animals were allowed to raise their own calves and recover naturally. They were then bred by artificial insemination in the end of April. We used our synchronization program on them. And then we determined pregnancy around the end of May. These animals were turned out by a bull until June or July, late June, early July, depending on where they were in the herd. And then we tested them in September for pregnancy by rectal palpation and a blood sample. So when we looked at their cow vitamin D levels, we significantly increased the amount of vitamin D by this injection routine. And this was just measured right before calving. So we elevated it into our therapeutic level. We wanted to hit about 50 nanograms per mil. Our controls were not bad with their vitamin D. They actually had this year between 30 and high 40s, depending on parity. So we were very happy with those data. And very excitingly, when we looked at the proportion of those females that were pregnant, here showing the black bars versus the proportion that were not pregnant by artificial insemination. Again, this was their first cycle, their first synchronized cycle. More of the treated animals were pregnant after artificial insemination. So they had presumably recovered to cycle better than our controls. Our controls were at what we expected, that we would be a little bit less than 50% pregnancy rate. So we were very excited by those data. When we looked at the calves, so we also looked at the calves. When we first checked calving ease, one concern was that we're treating with beta carotene and vitamin D. We might inadvertently trigger some excess growth in that calf. We did not want the calves to be excessively large at this point. When we looked at calving ease, and this is a scatter plot that my graduate student generated, where if you have a calving ease of 1, which is no or no assistance, up to 3, which was what we would classify as a hard pull. So this is kind of subjective. But we had an equal distribution of calving ease scores between control or treated cows and first calf heifers. And the body weight of calves also reflected this. So we split them out by parity too. We were a little concerned that we might have maybe affected our first calf heifers and maybe not our second calf offspring. And we did not see any differences in body weight of the calves at birth. So we were very excited again by those data. We didn't see any negative effects. But we saw an interesting effect in the calf serum vitamin D levels. So at birth, shown here in the yellow, are vitamin A and D injected. So the calves whose mothers were vitamin A and D injected during gestation, they had higher vitamin D levels than our controls. However, we tested them again 1 month after birth. And we chose 1 month because all of the data suggested that these minerals, both vitamin D and vitamin A, should have been stored for 2 to 4 months in the liver. Again, those data were from mostly steers and lactating dairy cows. And we measured the vitamin D levels of those calves at a month old. We no longer saw a difference between treatment group. Those calves that were treated had vitamin D levels at 1 month of age that were not different from our controls. So this was really interesting to us. We still are learning a lot about different rate of clearance of these vitamins between the calves. And so this is actually something that we are now going to follow up on further. So we are going to, as our previous presenter talked about, we are actually going to be collecting some placentas to start looking to see if we're affecting the placenta at all in its concentration of the either vitamin A active form, that retinol, or the vitamin D. We're going to look to see if there's any changes to the placenta that could be beneficial or even detrimental to the calf. We do not know at this point. And we are going to potentially in the future follow up on the calves and see maybe if we continue to treat them, what effects they might have. So we are still continuing to study this. This is still a really active area of research in our group, but we have some really exciting data. We think we have started an oral supplementation. So we are going back to that idea to try and see if we can use an oral supplement instead of monthly injections, because that is going to be something that's a little more translatable to a rancher out here in Oregon. So a lot of ours like to, as I'm sure across the country, some of you are more familiar with, having those animals out on range. And not being able to bring them up every month for an injection. So stay tuned. Those studies are still ongoing. We need a lot of animals for those studies because of the variation in how much these cows eat. So with that, also acknowledging in our research group, I have a lot of students that help with our projects. My students are amazing. They're incredibly enthusiastic. They love working with the cows. So I have some pictures of some of them here. The individual in front where we actually are all cleaned up and not in cows, this is the graduate student, Amanda Fox, that has been working on all of these projects as part of her degree program. So with that, I would like to say thank you for your attention, and I look forward to any questions.
Thank you, Dr. Bishop. That was an amazing presentation. It's always good to get a refresher of all the repro details and stuff. I myself kind of have to remind me all the time about those things because I'm more on the nutrition side and, all the relationship and, for repro to function properly needs good nutrition. But if we don't think about all those details, and it's very complex. So
Yeah.
I appreciate the reminder, and I think we probably talked about this before, but I really appreciate your work in vitamin. I recently started working with vitamin too, more specifically vitamin B, and it's so challenging, so complex. Mineral, we're always challenging. Vitamins is another level of complexity right here. So it's nice to see other people doing that work and also seeing positive outcomes like your preg rates and everything. So very nice.
Thank you. Yeah, no, and with these mineral supplements, it's pretty, straightforward. That mineral goes in, that's the one that's going to do it. With these vitamins, they're going to get converted to other things. So we have to be really careful about what are we studying and what are we actually seeing.
Yeah, no, that's very important. We do have a question on the Q&A box. If you want to go ahead and read that for us, and then
Yeah. So yeah, Jocelyn has a question on, are there any disadvantages to synchronizing a herd? So the only thing that I have ever seen as a disadvantage is if you start synchronizing animals that are not prepared to be synchronized. So if you are trying to synchronize those animals that are a little less mature, that have the lower maturity, you're not going to see as much benefit from it. So by waiting until those animals are a little bit more mature, you're going to get a bigger bang for your buck. So you might be spending money on animals that are not quite ready for the synchronization. Now, that being said, if you're willing to use a little more fancy synchronization protocol, you might kick some of those into being prepared and mature sooner, but there is always that cost benefit. What is the ideal starting point for breeding heifers in terms of age, weight, or body conditions? So yes, so that age, and I will admit that age is for Angus, an Angus breed, because that is what I am most familiar with. We like that 398 day, 300, right around 400 days. Those are going to be yours, that are going to be so nice. They're going to be ready to go. They're going to get pregnant earlier. They're going to calve and have enough time to recover before that next breeding season. And again, that body condition score, if you make sure you kind of hit the sweet spot, I don't usually look at weight. I look at body condition score because weight is going to vary a lot by breed. But if you get that body condition score, and so that ideal body condition in a beef cow is about a 5. So I like to put my hands on them too, not only to see them. I want to put my hands on them. I want them to be fleshed out. I don't want them to be too obese. I want to be able to not see a tail head, but I want to be able to feel it easily. So thank you, Erica. That's a great question. What's the source of the vitamins used, and are they protected? So we actually used just commercially available vitamin supplements. So these are commercially available. We chose those that did not have a lot of other co-supplements with them. So yes, these are just commercially available. The vitamin D supplement that you can grab at your local, and again, I'm going to use the store we have here. You can go down to Coastal, grab it off the shelf. But you got to be really careful with the dosing. And again, the dosing, a lot of the dosing on those is for an animal that is in a deficiency situation. So keep an eye on the space. We are still investigating different dosages and different responses, but always keep that in mind when you're grabbing those injectables. Yes, we're using them for research because that way I know that cow is getting the dose of vitamin D that I want her to get. But, don't just read the dose on the label and think that that's what all of them are going to need. So that dose is for a deficient animal. Very great question. And that's for with the oral supplements, you're going to want to work with a really good nutrition company. You're going to tell them, hey, I want a supplement with vitamin D, and they're going to help walk you through that process. So great question. Are there any correlations between vitamin and mineral deficiencies regionally and how rebreeding success? Yeah, breed back rates better in Florida versus North Dakota due to higher vitamin D. So that is a really, really great question. Do we see differences in breed back rates? I'm going to say that there's a lot of different data. There's a lot of different individuals that would say they have great breed back rates and they're in North Dakota in the middle of winter, and that's but they may have supplemented those animals. They may have prepared them really well. Unfortunately, because vitamin D and some of these other vitamins, as Juliana pointed out, we are just now beginning to dive into them, especially in our beef cows. We just assume they kind of all got that in their feed according to the NRCs. So I really look forward to the NRCs being revised as soon as a lot of us get these different data out there. But that is a great question.
Yeah, that was great. I was just trying to think about it. If there's any, there's one, you talk about the calving school, right? And how we offer those calving schools. And this is definitely one of probably my favorite programs here in Oregon. For those who are not familiar, we offer calving schools where we talk about the calving process, and we help producers to understand the calving process and help them to deliver a calf in dystocia. So I think it's a really cool program. And part of that program, we do like one of the slides, there's one study we talk about the calf size difference at birth due to location. So they had the switch of the herds from Florida to Montana. That made me think about the nutritional, like the vitamins and minerals, but I don't know if anyone has ever done that to that level. It's interesting. Very, very good question. Made me think about it anyway.
Yeah, no, that's a great question. And yeah, and it really is something. So even that group that did that big study, I have talked with them before and quite a bit about their study, and they've never followed up on it. They
Oh,
just constantly supplement their animals with oral feed with vitamin D in there, and they've never followed up on it. And so that's what I'd be interested in. I'm like, why aren't you guys following up on it?
yeah,
But now they're just doing this.
yeah.
So there is one other question. And this one, I'm going to toss this one to our beef expert here because you have captive audience. What is a good calving ease direct CED number?
I think we can go with a 5 or 6 if we're talking about heifers. That would be my go-to number. But like I said, that Reaper is not such my thing. I'd probably do a little bit more research and get back to it. But I think that would be my recommendation.
Yeah, I like to go with it. If I can, if I've got a good bull with other numbers and he has a 6, I like him.
Yeah. Okay. Well, I think that's basically all the questions we had at this point. I will offer to attendees that if we do have questions later, feel free to email me and I'll pass along to Cecily or to Anna if you were here for her presentation earlier today. But if we don't have any more questions, I would like to go ahead and do our door prize drawing. So Cecily, we have 37 people, 37 attendees. If you can pick a number, just a random number. From
Random number 12.
12. So let me do my counting here. Kelly Roney, you are our winner of tonight. And I'll reach out to you by email to confirm your information to send the gift card. But I think with that, that's all we have for tonight. Thank you very much, Cecily, and I hope to see you all tomorrow for the third night of RancHER. We will have Carlin Patterson and Sam Worth talking about trace mineral nutrition in dairy cows this time, and we're also going to be talking a little bit about beef production sustainability. So I hope to see you all tomorrow. Have a good night. Thank you, Cecily.
Thank you, Juliana. Bye.
Bye.